So hello students and welcome to the week 5 of the next generation sequencing class. Um, this is really the beginning of the course where you'll want to pay special attention owing to the fact that this is the portion of the class where we'll be talking about the SNP calling pipeline that some of you anyways, will be developing for your course project, the final project for the class. Um, both over in this series of lectures in week 5 and the lectures from week 6, I'll be outlining the details you need to implement the SNP calling pipeline. And just as a side, I've started working on a handout that will sort of guide you through the process and help you to know what the requirements are for the project. I'll be handing that out next week, and we'll be discussing that more in the webinars. So what do I mean by a SNP calling pipeline? So just briefly, what I'll be outlining today is the process for how to go from raw data, that is the FASTQs, coming off of a standard next generation sequencer, all the way through to the SNP calling step. The SNP calling will be reserved mostly for week 6, but all of the alignments and generation of the alignments and the processing of the aligned reads in preparation for SNP calling is really the subject of this lecture. So just to review what FASTQ format is, this was from week 1. Typically it's a 4-line per read format, where we have the forward reads in one file, the reverse reads in a second file from a paired end run, that is. If you have a single end run, you end up with only one FASTQ file. The reads, remember, are oriented such that pairs correspond to the same lines of the file. So if you have a forward file, the first read in the forward file will be, say, read 1. Its corresponding reverse read will be in file 2 for the reverse reads, also sort of beginning at the first line of the file. So all of the software that uses the FASTQ formatted files, remember, has no way of connecting based on sequence identifier the reads from file 1 and file 2. It's simply using the order of those reads, which the sequence or the pipeline that processes the sequencing data and exports FASTQs must get that right. And for all of the standard sequencers, we can assume that the reads will be oriented or sort of positioned in the right positions in the forward and reverse files. Anyways, that's just sort of a note, because that was something I worried about when I first started handling this data. So I'll let you go back and review what these different lines mean. I just thought I'd point out that there is no real specification for the identifier in a FASTQ. That is, the identifier can or may not incorporate important information. In Cassava processing pipeline, this is the software used to process the raw image files that come from a Illumina HiSeq run. There is important information in the read identifiers that's actually used in the processing pipelines that we'll be discussing. So in particular, I've colored in red, from the Cassava spec here, the positions of the read name that are important. The tile number tells where on the flow cell the read is coming from, okay, sort of as a region. The X and Y positions of the individual cluster in that region. Yes or no here, Y meaning yes, it passed filter. That is the default sort of Cassava processing filter. And also contains information about the barcode. So this is, remember, when you have multiple libraries in a particular lane, you end up sequencing a barcode so that you can distinguish them. Actually, it's Cassava that distinguishes the different libraries using this barcode and actually processes those libraries so that your FASTQ should only contain the reads from one barcoded library. Okay, the FASTQ should not contain reads from multiple libraries. So we also reviewed in week 1 the SAM BAM format, we'll go into this in detail in a second. Just remember that SAM, this is a raw text alignment format, which is really just the text converted information from a binary encoded alignment. A binary allows the information to be extracted much more rapidly by these software tools that we'll be using, and whereas SAM allows you to go in and sort of as a human readable form that we generally don't work with, but when we're trying to understand what's going on in a particular alignment, we often will work with SAM, and that'll become clearer as we go along. This is really just a cartoon I used in week 1 to introduce the BAM format, and we'll now be going on into much more detail, and you'll also be sort of encountering this in your homework assignment. So this is directly from the SAM specification. That is sort of provided as a supplementary material in this week's course content on the website, the Blackboard website, that is. And these are in SAM format. Here are the columns. SAM is an 11 plus column format. Just very quickly going down, through the 11 columns, we have the query name. That is the name of the read. The bitwise flag, I'll explain that in a moment. The reference sequence name is the name of the chromosome or contig that a read mapped to. The position is the coordinate that read mapped to on that chromosome. Map quality, this is a Fred scaled mapping, error probability that the read is mapped correctly. The cigar string describes the individual pattern of the base matching, and describes the alignment itself. The RNext column describes actually the information of a paired read, that is the mate of the focal read. So I'll be using the term focal read to describe this query name. So admittedly, some of this terminology, the reason I'm going through this with you, some of this terminology is a bit cryptic, I sort of admit. It's adopted to maximize sort of the unambiguity of the format spec, and yet just explaining it, sometimes we don't use these same terminology, so I'm kind of deconvoluting that for you here. But RNext, anyways, is is actually the name of the chromosome or the contig that the paired read or the mate of the focal read map to. So the reads are named the same. That is, the focal and its mate are named the same. So there's no need to actually repeat the name of the read here. The pnext, that's the position of the mate. The tlen here is the distance between the paired reads on the chromosome. The sequence of the focal read and the Fred scaled base quality plus 33. This is directly from the FASTQ, which contains the base qualities of the individual nucleotide bases in the sequence. So here's an example of what a single record or entry from a single line in a SAM files. Excuse the presentation here where we have multiple lines and problems wrapping here just to illustrate. But in any event, we have the read name, we have the bitwise flag, which I'll be explaining in detail. 2L, this must be coming from Drosophila, which the short arm of chromosome 2 is named 2L. 4 here is that is the position, so the coordinate, that's base pair position 4 on chromosome 2L. This is the mapping quality, the cigar string. Equals here is RNext. Okay, that just means that the chromosome that the second read or the mate is the same chromosome. It's equal to the 2L. 247, that is the coordinate position, the distance between, this is the T-length, the distance between the two reads, and this is the base sequence, the base qualities. Now these tags I also wanted to mention briefly, in particular that these are not absolutely required, but they contain, in many circumstances, important information, such as the read group tag. This refers to a specific line in the header which contains information about sample 44L1, including, what sequencing this read came from, so a HiSeq or a solid or some other sequencer, what library it came from. So in human sequencing, typically or frequently anyways, you're sequencing across multiple libraries for the same individual, or in other cases you're sequencing from different lanes. The whole sequencing strategy is important in some cases to interpreting the output data, and this read group helps you to know something about the sequencing strategy without having to report all of that information for each read in a very large file, right? So instead you just refer to the read group, which is in the header, which explains all of that information for this particular read. Okay, these some of these we'll encounter later, but mostly this is just details here that you can follow up on your own. The bitwise flag column, I did want to spend a moment simply because it's not intuitive, and I do have you go through it on the homework a little bit. I take you through in sort of excruciating detail in the homework about how to interpret these bitwise flag, but I at least thought I'd mention it here. First of all, notice here that the column 2 will have an integer. It is a decimal formatted number, okay, that contains a lot more information than actually you might think, just by looking at it. So here's a 163. So what does that 163 there in that bitwise flag position of the SAM format mean? Well, first of all, we need to convert that number into binary to make sense of it. Okay, so we have a base 10 formatted number. That's the typical numeral system that we work with on a day-to-day basis, but computers work on binary. But the reason really there's two reasons why this binary system works so well. First of all, the overall principle here is that these binary numbers contain important information about properties of individual reads. Okay, so that number, as I'll explain in a second, tells you something about that particular read. Second of all, since computers operate in binary, that is, they work on individual bits, this provides a means by which computers can work on and identify reads of certain classes or that have certain properties very quickly. So let's go through an example here. This is 163. That was from the previous slide. If we convert that to binary, we end up with this number. This is 163, which is a decimal number in sort of the binary representation. And each one of these columns, going from right to left, has a meaning. And these are sort of Boolean characters here. That is, they mean 0 or 1. 1 typically means true, 0 means false. And so here, for just as a way of example, column 1, that is working from right to left, okay, column 1 here is whether or not the read was paired in a sequencing run. Okay, so did it come from a paired end run? 0 would mean no, that would be a single end run. 1 means paired end. Okay, I'll let you go through some of these details because you do that in the homework, but each of these columns moving from right to left has an important meaning, or a specific meaning, that is, and really these are all Booleans, that is, true or false. Does this read meet that condition? So hopefully you can keep that in mind. I interpret the full the binary, it's sort of the interpretation of this binary here for the value 163. So I don't expect you for this class to be able to convert decimal to binary. Okay, so one thing that you can do, if that seems a bit cryptic, or you don't have any computer science background, perhaps you can go to this particular website to sort of explain these bitwise SAM flags for you. So let me just quickly take you to that website and give an example. Let's put in 163 here and then click explain. And what it does is it tells you, all of the information that you need to know sort of in a human readable format as to what the number 163 means. Okay, we can also sort of uncheck these or delete these, and we can say, okay, what if we want to know what digit or decimal representation would be for, say, a read that comes from a paired end library, okay, that is unmapped. So the read is unmapped and it comes from a paired end library. Well, that value would by itself be a 5. Okay, notice that these same properties, okay, let's say that we then say, what if we want the read to be unmapped, but we want its mate unmapped also? Notice how the number changes. So you can't simply identify all reads that are paired in the run and are unmapped by extracting the records from the SAM that contain a 5, because values with a 13 also meet those conditions, but they also meet additional conditions. Have you sort of like go through and think about this a little bit in your homework, what that means, but you can't simply work with that integer value in column 2. That is not how you extract reads with certain information from a BAM. With certain properties. You cannot extract reads from certain properties by simply, say, grepping on a certain integer value in column 2. You must use SAM tools in order to pull out reads that meet certain conditions, and I'll show you how to do that. Okay, so one other thing I wanted to point out here is that this sort of was not, you have to read the SAM specification very carefully to get this point, but a read is if it's marked as unmapped in the bitwise flag, then you actually can't trust, or you can't use information about from these other columns. Okay, so the first thing you always do is to check whether a read is mapped or not. If it's not mapped, then you can't make any assumptions about these values. In fact, these values may be misleading or nonsensical in some cases. You must first check to see whether a read is mapped or not. Okay, so the map queue is Fred scaled, so we can apply the same formula into determining the error probability of a particular mapping. And the map queue of 0 often has a special meaning, which means that the read itself is multiply mapped. Okay, so let's go ahead and look at some FastQC data here. I just really, I'm taking you back now for review. You might remember this figure from the week 1 lecture, but I also wanted to point out here that this figure was generated by a toolkit called FastQC. FastQC provides a means for summarizing the raw FastQ data. It gives a sort of human readable quality reports that allow you to identify coarse problems with a FastQ run. The results are reported in easy to read HTML format for ease of viewing by non-expert viewers. So I present this to you here only. We won't have time in this class to go into any more detail. This is a very simple thing to run. I didn't want you to have to go through the process of installing everything just to sort of extract this to be able to run FastQC. I simply wanted to point out that as a bioinformatician, something that you might want to build into your processing pipelines is a FastQC step that will allow you to sort of visualize some outputs that give you some sense of whether or not your data are good or not. So let's go over what some of the generic steps for read alignment, alignment processing, and SNP calling are. So the first thing before you can do anything is that we need to create what are called indexes of the reference genome. Okay, so I think I go in a second and tell you why we need indices. I'll just mention it briefly here, is that essentially indices allow a software to go in and jump between positions of the file. Most scripting languages require that you go line by line. So if you know Perl, for example, when Perl reads through a file, it has to go line by line to read the information from the file. However, that's very slow in working with very large files, and so the references allow a program to jump to a particular coordinate position in a file, which therefore greatly increases access time. So BWA index and FASTA index, we'll be talking about that some more later. These are two that are requirements in order to do alignment, and FASTA indices are required for the vast majority of third-party tools. Part of this generic steps here is to align the reads. These are the short read aligners that we'll talk about in a second. We need to fix made information. I'll explain that. We need to coordinate sort our reads. We need to mark duplicates. I'll explain that. Finally, we have to do what's called a realignment around insertions and deletions. This has the requirement for this, as I'll explain, has to do with the high false positive rate due to misalignment of reads around regions that are inserted in one genome relative to another. We then finally need to merge the sample BAMs, and as I describe next week, you actually do SNP calling typically on many samples, that is, many individual genomes simultaneously in order to reduce the false positive rate in the SNP calling protocol. And then finally we'll talk about the SNP calling protocols themselves. There's also a couple of advanced steps. We probably will revisit this briefly next week. So the first step here, let's go back here to the SAM and BAM alignment. So how do we generate these alignment files? So there's many short read aligners available, so that will convert a FASTQ into a SAM and BAM by aligning to a reference genome. They differ based on algorithmic details that are sort of beyond the scope of this class. These algorithms differ in speed, the optimality of the alignments, whether they find a true optimal alignment or not, and whether the alignments are gapped or not. That is, whether there are gaps. That is, insertions, deletions. So some of the common examples here are Burroughs-Wheeler aligner. This is what was used by the 1000 Genomes Project, and the aligner we'll be focusing on here in the class. Bowtie 2, we'll use this for our RNA-seq down the road, but for DNA sequencing it's used a little bit less because it's fast but less accurate. Stampy, just one that I know of because colleagues are use it for very cases where you're looking for single mutations or something where you really need to find the absolute most optimal alignment. Shrimp is one that I know of that's used for solid or color space data, which we won't be talking about in any detail in this class. The properties of desirable aligners, at this point, all aligners really that are worth their salt are output gapped alignments, that contains insertions and deletions. These alignments should be in SAM or BAM. They should handle paired end data, and they really should be sort of optimally suited for your needs. So what I mean by the trade-off between accuracy and speed should meet your experimental design is when we're doing RNA-seq, we'd like to map quickly the reads to the reference, which allows us to sort of speed up our experimental design. Excuse me, we'll probably be using top hat. I'm think I'm misspeaking here from this package rather than bowtie 2. But in any, when we do the RNA-seq alignments, but in any event, bowtie 2 is fast, less accurate certainly than BWA or Stampy. But so we use BWA because we're in many of our analyses, as did the Thousand Genomes Project, because it provides a reasonable trade-off between speed and accuracy. Whereas Stampy is very, very slow, and is simply too slow to process large volumes of data. So again, BWA, the Burroughs-Wheeler transformation. It requires 3 steps. I'm pointing this out in some detail here because we may not encounter this. There's not a convenient place to discuss this in more detail, and so you might want to pay attention to this if you're planning to perform for your project the SNP calling pipeline. So BWA index, BWA align, BWA SAMPy. We first have to index the reference genome. We align the reads individually, sort of ignoring the paired end status of a pair of mated reads. And then BWA SAMPy goes through and then connects those paired ends. You can read this about this in some detail, but these are really the 3 commands that you must execute in your pipeline in order to get BW to work properly. And I'll let you read the BWA documentation to learn how to implement these 2 programs. So this is here the sort of the program package, and then so BWAs and SAM tools were both written by Hang Lee. So you'll see a similar format here where you have the name of the sort of program package, and then these are individual programs within that package. So for the BWA SAMPy step, you redirect the output using these standard by redirecting the standard output to a file name .sam followed by a SAM to BAM conversion, which you'll learn how to do in your homework. Okay, so keep this in mind. You can come back and review this as you're developing a strategy to complete your SNP calling pipeline. So I thought I'd touch on this briefly. This is a bit of details, but it is necessary when you're the bioinformatician and you have to get these things right. So curiously, BWA and other aligners sort of don't output perfectly formatted BAMs. They've actually output BAMs that have errant information in them about the mate. So if I may, maybe the easiest way to sort of explain this is to go back to the SAM-BAM format, and let me point out something explicitly. There is redundant information in BAM files because a BAM contains in one record, okay, or one line of the file, it contains the information for a focal read and its mate, okay? Its mate's information is here. So a second line of the BAM file, will contain this other read, that is the mate of the focal read, will be the focal read in another record, and the mate here will, appear as rnext and pnext, or its information will be in rnext and pnext. So the rnext and pnext contains information that's found twice, okay, in the file. That is once here where it is not the focal read, and a second time where the chromosome that this read mapped to will be found in the R name position of the SAM file. So the point is we have redundant information here, and that information can get messed up. So in other words, a read may not actually have the appropriate position for the mate, okay, and it may conflict across the 2 places in the BAM file where that information is recorded. So what fixmates does is it assumes, as it should, that the focal read information is correct, but in cases where the focal read information differs from the same information in the mated read, then what happens is that the fixed mate information will go through and it will correct that by extracting the focal read information of the R name and the positions data and going to the mated read and fixing in that information in the R next and P next. If you have questions about this, let me know. I think that's fairly straightforward. Simply that there's redundant information that can get messed up, and other than that, you just need to be aware that we have to go through and run fix-it mate information after we run our aligner. Okay, this is a Picard Tools protocol. So you'll be installing Picard Tools, when you go ahead and proceed with developing your snip calling pipeline, and you will need to run the fixed mate information program within the Picard Tools package. Okay, so this is all pretty much what I just explained to you, and so I'll let you read about this maybe in some more detail by looking at the documentation in Picard Tools, but this is essentially why you need to run fixed mate information. You also need to sort SAM and BAM alignments. So an alignment can either be unsorted, coordinate sorted, or read name sorted. That is, sorted based upon the identifier. From a practical perspective, sort SAM in the Picard Tools package is the way to go. Rather than using SAM Tools, which also has a sort function, I highly recommend using sort SAM. It's memory intensive, creates a lot of temporary files. So when you're coordinate sorting a BAM, that is, you want to get it so that the all of the reads appear in the order as they do on their mapped chromosomes. That is extremely memory intensive, and so there's so much memory required that it has to dump the data to temporary files, and sometimes this is a difficult step to get to work. So if you are running into problems, of course I'm going to be giving you a data set that's far reduced in terms of the numbers of reads, but just as heads up, if you're trying to develop this, you may encounter problems here in SortSam. I certainly have in the past. So how do you know if a BAM is coordinate sorted or how it's sorted? Well, the idea is to actually, you can write some code to check column 4, I believe it is, to see if the coordinates sort of appear at least within a chromosome, from in ascending order as they should if it's coordinate sorted. But a shortcut is to look at the BAM header. I haven't described to you too much about the BAM header at this point. We'll look at that a little bit in the homework, but before you get to the read section in the BAM file, there's a header section. I explained this a bit in week 1. That has important information, and I described the read group information earlier. But there's one line that has this HD indicator here at the beginning of the line, and the SO tag. The SO tag will tell you if it's coordinate sorted or not. And I will leave that to you in the homework to go in and check to see whether the BAM file that I give you in the homework is in fact coordinate sorted. Alright, so we've talked now about the alignment protocol, that we need to fix mates after we do the alignment, that we need to sort on the coordinate sort our BAM. And then the next thing we need to do is mark duplicates. And so before telling you practically how you go about doing that, I thought I'd give you conceptually what is a duplicate and why is it a problem for snip calling. So let's imagine here a set of reads. Okay, so this is a forward read, this is a reverse read on a mapped onto a reference genome, and the red lines here indicate positions where the 5' end of this read, of all of these reads here, the first, let's see, 6 reads on the left here, all map to the exact same position. On the right here is reads on the reverse strand that also map to the exact same position, that is these 5 reads here. And the ones I want to draw your attention to are the reads in the blue box. These are what we call PCR duplicates. So a read, this means the fact that these reads map to the exact same positions, okay, means that they come from a fragment that was randomly sheared at the exact same positions. Now, it is possible that these are not PCR duplicates and that they come from two independently sheared fragments from the original genomic DNA preparation when we went into preparing the libraries. That we happen to sequence, totally independently. That is possible, but it's highly, highly unlikely, okay, unless we're sequencing at extremely deep coverage. So more likely is that a single molecule of DNA, okay, that was sheared off here at the positions indicated by the two red lines, was then amplified during what we call the polymerase chain reaction step. And that is actually the same molecule being sequenced multiple times. Now, generally speaking, that, or naively speaking anyways, that you might not think that that's a problem. Why is sequencing the exact same molecule a problem? Well, the problem here has to do with the fact that PCR leads to a high error rate, okay, such that a single molecule has a has a finite probability of it having a mistake relative to the template DNA that was being replicated, okay, or amplified. And those mistakes end up, if you look here, let's imagine the red X is a G where the correct sequence is an A. Okay, so the correct nucleotide is an A. These are PCR errors. Okay, so incorporated by a biochemical misincorporation of the wrong base during an amplification step during the data, sort of the initial steps of library preparation. And that what does that look like? Well, that looks like a SNP. Okay, it looks like there's an AG polymorphism here, and maybe the individual that's sequenced is a heterozygote. So that is a problem, and we need to try to remove that. So the way that marked duplicates identifies this class of duplicate is it looks for pairs of reads that map to the exact same position. Once again, we will occasionally be tossing out reads that are actually perfectly legitimate because they come from two independent original fragments that may not have been amplified successfully at all during the PCR step, which in that case, this situation wouldn't be a problem. But in the event that these are amplified products that represent a single template DNA in the original genomic DNA preparation, then this is a problem. So I'll let you think about that. Ask questions. Feel free to ask questions if that doesn't make sense. But just to point out here that this cannot be a duplicate, okay? Because a PCR duplicate anyway. Because this pair of reads, while the left end maps to the same position as this problematic set of reads, the right end of the pair does not, and so therefore can't come from the same original fragment. Same for this pair of reads here. So another class of duplicates has to do with an imaging problem, on the flow cell, where again we don't want to, be, assigning or attributing reads that come from the same PCR product as being independent original genomic fragments. So another way in which you can sort of be analyzing reads that come from the same PCR or fit from the same genomic fragment but we're sequencing multiple times, or they appear multiple times in your alignment as different read pairs, is because of an imaging problem associated with the imager and detecting clusters on the flow cell. So each color here represents a different cluster, But in boxes is how I've been, I'm just illustrating um, how the imaging algorithm breaks up the clusters and detects them as being separate. So here, the light blue, I'm showing, or trying to illustrate that this cluster, which is sort of growing like an amoeba, as it's proliferating on the flow cell, can be interpreted as actually being part of two clusters. Okay, here, whereas this one is interpreted correctly as being one cluster. Okay, and then we could draw boxes around each of these individual clusters. If we wanted to, flesh out the figure, but the boxes should be around one color, but here we have two boxes around the same color. And so what that could do is this can artificially be interpreted as different, genomic fragments when in fact they are coming from the same fragment, and this can lead to similar problems to the PCR duplicate problem. I don't expect you to learn, to know sort of all of this rationale in any, detail. Just be aware that it's a problem, and actually, this is the case where we can fish out duplicates that are what we call optical duplicates because their coordinate positions on the flow cell, okay, are listed in the read identifiers, which I pointed out at the beginning of the lecture. And so we can fish out reads that are close to each other on the flow cell, okay, and in that way we can, and those reads map to the same position, we can identify them as being duplicates or optical duplicates. Okay, so that in the end, the goal of marking duplicates is to reduce the false positive rate associated with calling SNPs. When we have these, artifactual situations introducing potential biases. Okay, so I've explained what a PCR duplicate is, why duplicates are a problem, how to handle duplicates in BAM files. We mark or remove duplicates in the BAM. We generally have that option. What I'm asking you to do in your alignment program, alignment pipeline, is to simply mark the duplicates. And so, as you may have noticed in that bitwise flag column in the BAM, this is where the so-called marking takes place. Of course, you don't have to do that by hand. It's just that information is recorded in the bitwise flag by the Picard Tools Mark Duplicates program or SAMTools RMDup. For reasons I won't go into, if you do some, digging around, you'll find that even Hang Lee himself, the developer of SAMTools, recommends using marked duplicates for reasons I won't go into. So this is the standard now way of marking duplicates in your BAM file, and that will be something that you'll need to incorporate in your SNP calling pipeline. Single command line. All of these are single command lines for all of these steps that I'm describing to you are break down to essentially single command lines that you just have to get the command lines correct and in the right order, and you'll have developed your pipeline. So it's not complicated. I think you'll be able to do the project fairly quickly once you sort of grasp things, and of course I'm here to help, and I also don't mind if you work with your other members of the class if you wish to as you're working through your pipeline. So the thing I want to close with, because this lecture is getting a bit long, it's obviously rich in content that's of value, I think. This it really is some of the basic analyses that I think any bioinformatician should be able to do. Although pop quiz, and I'll bet you many might not get these steps right, as this requires some digging to finally arrive at what the correct commands are and the sequence of events that need to take place to come up with the best alignment files that you possibly can have for SNP calling. In any event, The next step, and the final one we'll talk about today, is the realignment of reads in insertion deletion regions. So here what I'm showing you is, if you can see it, is in blue here are individual reads. This is an insertion deletion. That is, there's a region here that's in the reference genome that the sample genome, the one being sequenced, which the reads correspond to, does not have any sequence there. Okay, the problem is that reads can shift to the left or right because of the simplicity of the nucleotide code, A's, T's, G's, and C's, and can get misaligned. And I don't know, this part you probably can't see. Take my word for it. There is what looks like a G, alternate base here. That's being called, that looks like a heterozygote, and there is an A here, alternate base, right next to it, that looks like a heterozygote. And yet, this is before the indel realignment. Then when you realign the indels, everything sort of shifts about one or two bases to the left. Notice an additional sort of deletion relative to the reference appears here in a larger fraction of the reads. But the upshot is that the SNP goes away. This pair of SNPs adjacent to each other goes away when you simply shift the alignment one or two base pairs. And so this is the problem that the realignment around insertion deletion polymorphisms is meant to deal with, because these are false positive SNPs, which are now gone. Okay, with that, I'd like to close this lecture. Tune in to the second lecture, the week 5, for additional details for your SNP calling pipeline.